Hello and welcome. This is the fibrous proteins, one of the two fibrous proteins videos. After this video, you should be able to describe the general themes for all fibrous proteins, the things that can be applied to all of them. Describe how structure dictates function for collagen, so that's a really big theme across biology, chemistry, and of course biochemistry. Is that structure dictates function, and so for a particular protein we'll do collagen, you should be able to describe how structure dictates function and how if you change that structure, a prediction on how the function itself will also change. And then compare and contrast fibrous proteins with globular proteins. Alright, here we go. So I've used this word globular a few times in class and some other videos, and these are the proteins that are water soluble, and that means they're, like this one right here, they're hydrophilic on the outside. This is where all their positive and minus amino acids are, and the polar neutrals. And then they're hydrophobic on the core. So their structure is driven by, their three-dimensional tertiary and quaternary structure is driven by the hydrophobic effect. Now, fibrous proteins are a little bit different. These ones are insoluble, so they would not be soluble in water. They would crash out of water, and two examples of those are collagen and keratin, and here is an example of keratin. Hopefully you can see some general differences between them. Keratin is sort of this long structure, and hexokinase over here is almost a globular or global or spherical kind of shape. Okay, so here are the general themes for fibrous proteins. And what I mean by general themes is I mean that all fibrous proteins contain or comprise of these same three things. I don't know, they call them themes. I'm not exactly sure what is an all-encompassing word for them, but the first one is that they all are or contain a single repeating secondary structure. So that means that basically they're either all helical, the entire thing, or they're all beta sheet. In collagen and keratin, they're both completely helical. They're not even alpha helices, they're different helices, but they're entirely helical, sort of like how we did in class where I had the pool noodles and the pipe cleaners, and I had the students connect the pool noodles end to end, and that would be an example of an all helical or all secondary structure protein. All right, so all fibrous proteins have a single repeating secondary structure. The next one is that they are hydrophobic, which means that they're made of mostly all nonpolar amino acids. For example, a question to think about is, what happens when you spill water on your nails, on your fingernails? And your nails are made up of keratin, right? So if keratin was water soluble, your nails would dissolve when you spilled water on them, just like your hair, which is also made out of keratin, would dissolve when you took a shower. So that's how you know that your hair, your skin, your nails are made out of fibrous proteins, because they're not water soluble, they're hydrophobic, they repel the water. Okay, and then the third thing is that all of these are polymeric chains, so they're all polymers, they're all proteins, that pack together to form Supra. These are really big structures. Supra molecular structures. So we have lots and lots of strand packing here. Okay, so we're going to focus on collagen. We're not going to go through all the different kinds of fibrous proteins. We're not going to go through keratin and elastin. We're going to go through collagen because I think it makes a really interesting story, and it's just a really great example of a fibrous protein. Okay, so collagen is the most plentiful protein in your whole body. You have more collagen protein than any other protein in your body. It holds together connective tissues, tendons, blood vessels, your eyes. The way I like to think about this is it's so strong, that's its function. Its function is to be strong enough to hold your teeth in your gums or hold your eyeballs in your head. So that's its function, is to hold things in their place. So then you can see here is a super zoom out, and we keep zooming in and in and in. And this right here is what's called a Supra molecular structure. Molecular. I like this picture right here because calcium is bone cement. So if you add calcium plus collagen, you get a bone. Cool. So food for thought. And this is kind of going to be a theme throughout the rest of the lecture. Is like, what would happen if collagen wasn't so strong, or what biochemically could occur to make it not so strong? So if it wasn't so strong, your teeth would fall out. If it wasn't so strong, then you would have pain in your muscles and just overall weakness. So what would biochemically cause collagen? Well, what biochemically causes collagen to be strong? And then how could it be mutated to not be as strong? All right, so we're going to start with the primary sequence, and then we're going to go to secondary. We're going to skip over tertiary and then go to quaternary. All right, so it's a strand. Collagen's basic primary sequence is a strand that is a very specific sequence that has a glycine every third residue. So this is a strand going from N to C, where glycine is every third residue. X and Y are other amino acids, and they could be proline, hydroxyproline, and I'll draw that for you in a second, hydroxylysine, or alanine. If you look in this cartoon diagram, you'll see that you have a glycine that has no side chain, and then 1, 2, 3, there's another glycine, and then 1, 2, 3 is another glycine. So it's really important that glycine be the third residue, and then what's making up the residues in between is mostly proline and a hydroxyproline. And those are the ones that some of you keep saying look like a house. And yes, they do kind of look like a house. This would potentially be an alanine right there. Okay, so what the heck are hydroxyproline and proline? Maybe I'll stick in red here. So I'm going to start with my proline. These are all part of a primary sequence now, so I wouldn't draw the C terminus and I wouldn't draw the N terminus. Hydroxyproline is a post-translationally modified. That means after it comes out of the ribosome, it gets an OH. Hydroxy meaning OH, proline meaning proline, hydroxyproline. Hydroxylysine, NCC. My squigglies just represent that we're inside the polymer of the protein. All right, so the OH here comes on the third one down. H, CH2, NH3 plus. Okay, again, so we can abbreviate these HYLYS, that's hydroxylysine, and then pro for hydroxyproline. Again, these are post-translationally modified. That means the OH gets added on after the protein is synthesized by the ribosome during translation, post-translation, after translation. Okay, collagen is a triple helix. So you can see over here, I picked a rendering of spheres where you can see each of the helices. So each strand, there are 3 of them, that's a triple part. Each strand is its own helix. and then they wind together to make a big, giant helix. Okay, so it's a triple helix. It's a left-handed helix, which is a little bit bizarre. Okay, so let's talk about what it is about this structure that makes it so sturdy. Okay. Okay, so here's another view of the triple helix, which all the amino acids, side chains, and the backbone are all shown in sticks in CPK coloring, except for the carbon has been changed. So we're going to go through one by one, and we're going to talk about each of the amino acids, what its role is. So let's start with proline down here. So proline's role is to add a kink in the backbone. And it has a hydrophobic or non-polar side chain. Remember, that was a theme of the fibrous proteins, is they had to be hydrophobic. Okay, so what do I mean by kink? I mean that because proline's side chain comes back up to the backbone, that means in terms of torsion angles, it adds a sharp turn. So this helix, each helices is like a 310 helix, so there's a residue every 3, which makes it a tighter turn of a helix. And one way to achieve that is to add in a proline to get that sharp turn so that it turns every 3rd residue and not every 4th residue like in an alpha helix. Okay, hydroxyproline also adds a kink. A kink. It adds a kink. It's also mostly hydrophobic, but you get the added benefit of being able to have hydrogen bonding. And in the next slide, I'll show you why that becomes so important. Oh, I didn't even draw that proline right, did I? Yikes. Oh, sad. We're just going to get rid of you all together. Okay, so it adds a hydrogen bonding. So I want to remind you that with an OH, there are 3 hydrogen bonding opportunities that you can accept at each lone pair and donate at this hydrogen. So every hydroxyproline adds 3 hydrogen bonding opportunities, which is going to increase the strength of the structure because hydrogen bonds are one of the stronger non-covalent interactions. Okay, I'm going to combine alanine and glycine. So glycine is actually a little bit more important than alanine because it is so small. So its side chain is just a hydrogen. And alanine is the second smallest one with the methyl group. So the small side chains alleviate. Oh, I did not spell that right at all. Oof, that looks like alienate. Yikes, not even. Anyways, it alleviates the torsional. Why do I keep picking words that I can't spell? Torsional stress along the backbone. If the backbone was only prolines and hydroxyprolines, eventually it would either unfold or it would go to an unfold. I don't want to say it would break. It wouldn't break. It would unfold and would not come into a helical structure because the torsional strain, or we could also call it steric stress on it, is too much for it to handle. Now the one that's not on here that you can't see is the hydroxylysine. So I'm just going to kind of zoom out and put him off to the side because he's a little bit different than all the other ones. So hydroxylysine allows for covalent, so this would be super strong, covalent cross-linking between triple helices. So not within a triple helix, between triple helices. So if I had 1, 2, 3, okay, there's my triple helix, and I had a second one, 1, 2, 3. The hydroxylysines I'm just going to put an R here, allow for covalent cross-linking between them. So this would be one hydroxylysine, and this would be a second hydroxylysine. So that helps form, when multiples of these triple helices come together, you get your supra molecular structure. Okay, and I also forgot to add that not only do alanine and glycine alleviate that torsional strain from the kinks from the hydroxyprolines and the prolines, but the alanine, being a little bit bigger, is also hydrophobic, or nonpolar. Okay, so now you know all the amino acids that can be found in the primary sequence of collagen, and remember that glycine is every third. It's got to be every third, and if it's not a glycine every third, it's got to be an alanine, and that is super important for this. Okay, so looking at a couple different views, I'm going to kind of give you an idea of where these amino acid side chains are located relative to one another. Again, this is one chain that is itself a helix. It's a 310 helix roughly. It's actually a little bit smaller. It's 3 with 3 residues per turn. When you combine these together, this is its secondary structure. When you combine multiple chains together, you get to the quaternary structure, and you'll kind of notice that there's actually no tertiary structure here. We skip right into quaternary. So I really like this view right here. I think it's really interesting. So this is the top view. So looking down the helix, and what you'll see is on the outside, these are all the lysines. They have not become hydroxylysines yet. They have not been post-translationally modified. That's going to be in the next slide. The core here, where all this gray is, we call this the hydrophobic ring or core, is where the proline and hydroxyproline and alanine side chains are. So hopefully you can see some of these in here. You can see there's one right there. There's another one. So you can see all the hydrophobic parts, or the CH2s have all stacked on each other. So those are all in this innermost ring right here, that hydrophobic ring. Actually, it's not the innermost ring, it's a second layer in. So the lysines are the outermost, and then you have this hydrophobic ring, and then the very, very center, I don't know if you can see this, but this is where all the OHs, and this is where all the hydrogen bonding is going on. Okay, and the hydrogen bonding can be from the glycine or alanine backbones, not the side chains themselves, and the hydroxyproline side chains. Sorry, I should probably have said that could be from any of the backbones, but mostly it's glycine and alanine because they're so prevalent. So the innermost core of this, I don't know if this is hard to see with the colors, but where you can see these kind of oranges, those are supposed to be oxygen, so those should be more of a red color. That's where all of the hydrogen bonding is going on. So this centermost core is super, super stable with all the hydrogen bonding. And then, maybe I should, here I'm gonna erase my rings. Rings upon rings upon rings upon rings upon rings. And just walk you through it just a little bit better. Okay, so in the center core, the center of the triple helix, is all the hydrogen bonding, which makes the core super, super stable. And then the rings surrounding that are all the hydrophobic parts of the side chains that are stacking on each other. And the rings surrounding that are all the lysines that are sticking out. Now those lysines, like I said, covalently cross-link between triple helices. So here's just 2 lysines, and there's an enzyme right here, a lysyl oxidase, and you know it's an enzyme because it ends in ASE. Not all proteins are enzymes, and not all enzymes are proteins. but this particular enzyme is a protein, and it oxidizes, or in biochemistry that just means adds oxygens. So it actually removed the NH3 to add the oxygens, and then those were covalently cross-linked through an aldol condensation reaction, which I'm pretty sure you were introduced to in organic chemistry. But just so you know, no need to memorize this. Yeah, don't need to memorize this at all. I just wanted to show you a schematic diagram of how those lysines become covalently cross-linked. Now, the thing that's not on here is the hydroxylysine. So that arises, and I've searched far and wide, and there is no good picture of this. So the hydroxylysines, so it comes off of this one right here, and this one right here. I'm actually going to draw him on the other side. He wouldn't normally be set up quite like that, but... Oh no, they're on the third one. He's on the third one, not the fourth one. It's on the third one. 1, 2, 3. Yeah. He's on the third one. This one. And this one. So if these were in the right orientation and proximity to one another, then these two would hydrogen bond with each other without the covalent cross-linking. So not all the lysines will become covalently cross-linked. Some of them will have hydrogen bonding between the structure, and that actually allows it to be a little bit more dynamic. You don't want too much covalent cross-linking, because then your collagen gets really, really brittle. And if you think about collagen, and people get collagen fillers in their face and stuff, your collagen, one of its, other than being really strong, is it's very elastic. And so this hydrogen bonding here helps with the elasticity of your skin. So too much covalent cross-linking is not good. It makes your skin really brittle. So in order to keep these triple helices together, some of it is held together with hydrogen bonding. Kind of cool, huh? Okay, and then this last slide is just kind of like a food for thought. Again, you don't memorize this. Don't memorize. Don't memorize this at all. But this is basically, collagen is extracellular. That means that it exists outside of the cell. Well, how do you get something as big as collagen to assemble outside of the cell? You can't make the entire supramolecular structure, that's the supramolecular structure where the triple helices are coming together. You can't make it inside of the cell because it's just too big. Okay, so I think this one is really interesting because it shows that collagen is mostly synthesized by ribosomes on the ER. Okay. And then it gets glycosylated, or it gets sugars put on it. And those sugars help form small chunks of triple helices and help transport out of the cell. And then once it gets out of the cell, you can combine a lot of the triple helices together. I'll just say that one more time. So collagen, it's a protein, so it has to be translated by the ribosome. So it gets translated on the endoplasmic reticulum, or the ER. And then after that happens, and it gets glycosylated, or it gets these sugars put on it. And those sugars are useful for small assembly and also transport, like a signal, to get outside of the cell. And then once it's outside of the cell, they can form into these triple helices. I just thought this figure was really interesting, and I wanted you to think about, if collagen is extracellular and it's fibrous, meaning it's not soluble in water, then how does it actually even form? Because it has to start inside the cell, right? If it's a protein, that means it was synthesized from mRNA, right? It was synthesized from mRNA, and where does the mRNA come from? Well, it comes from DNA, and where's DNA housed? Was housed in the nucleus, and that's certainly not extracellular. Okay, these are just weird things that I think about. Anyways, I think that's all I have for you. I'm gonna stop talking now and let you do your homework, or maybe I'll just see you in class, and yeah, bye.